In this lesson, we're going to learn how the human brain speaks and communicates. Now, in order to understand how the brain works, there are two structures we need to become familiar with. The first is the neuron, and the second is the synapse. So let us start with the neuron. Just like all organs in your body, the brain is made up of cells, and the neuron is the name given to the specific type of cell that makes up the brain. In fact, your brain is made up of an estimated 100 billion neurons. Just like every other cell, neurons have a cell body. This is where all the machinery that keeps the cell alive is located. Structures that produce energy or dispose of waste, things like that. Now, neurons contain a number of additional structures that make them unique and different from any other type of cell. The first of these is called a dendrite. Dendrites are branch-like structures that jut out from the cell body and are made up of the same material as a cell body. With regards to communication, information comes into a neuron via the dendrite. Next, neurons have a long thick tube that shoots away from the cell body, and this is called the axon. Unlike dendrites, the axon is technically not part of the cell body, rather it's a wholly different and unique structure. So because of this, there's a name for where the cell body and the axon meet, and that part is called the axon hillock. With regards to communication, information flows along the axon. Now you might notice in this picture that the axon is occasionally wrapped in a kind of protective coating. This coating is called the myelin sheath, and this serves to insulate the axon, essentially separating portions of the axon from the external environment. The small areas of the axon that aren't insulated, that are exposed to the external environment, are called the nodes of Ranvier. And with regards to communication, we're going to see later what the purpose of the myelin sheath and the nodes of Ranvier are. Now finally, the axon ends by splitting into a number of small nodes called terminal buttons. Information exits the neuron via the terminal button. The second structure we need to become familiar with is the synapse. The synapse is where information exits the terminal button of one neuron and enters the dendrite of a second neuron. In the human brain, there are an estimated 100 trillion synapses. And if we zoom in real close to a synapse, we find that the two communicating neurons do not actually touch. Rather, there's a small gap between the end of one neuron and the start of the next. So within a synapse, we have three major structures. We have the presynaptic terminal, the synaptic cleft, and the postsynaptic terminal. The presynaptic terminal is the name given to the terminal button of the first neuron. So this is where information exits the first neuron, and within the presynaptic terminal are a number of small sacs filled with chemicals. These sacs are called vesicles, and the chemicals inside the vesicles are called neurotransmitters. Now, the small empty gap between the two neurons is called the synaptic cleft, and it's here where the neurotransmitters that allow neurons to communicate must flow across. And finally, we have the postsynaptic terminal. That's the name given to the dendrite of the second neuron. This is where information enters that second neuron. Now, lining the postsynaptic terminal are a number of structures called receptors. Okay, now that we're familiar with the neuron and the synapse, we can see how all these parts fit together to allow the brain to communicate. So let's begin by looking at a single neuron. Inside of each neuron are a number of ions floating around. Now, an ion is a molecule or particle that has gained or lost an electron. So this gives an ion a specific charge. Now, inside each neuron, there are quite a few negatively charged ions. Things like chloride, which is abbreviated Cl negative. That's how you know it's negative. Now, outside of each neuron, in the space surrounding the neuron itself, there are quite a few positively charged ions. Things like sodium, which is abbreviated Na+, so you can tell it's positive because of that plus. This means that typically it's far more negative inside of a neuron than it is outside of a neuron. And this difference is called polarization. So when a neuron is at rest, when it's not firing, polarization is typically around negative 70 millivolts. This means that there's about 70 millivolts more negativity inside of the neuron than there is outside of the neuron, and we call this value, this negative 70 millivolts, the resting potential of a neuron. So what happens to cause a neuron to fire? The process begins at that spot where the cell body and the axon meet the axon hillock. At the axon hillock, there's always a large number of ions floating and fluctuating around. So sometimes a lot of negative ions will gather at the axon hillock, thereby making the inside of the neuron even more negative than it typically is. This is called hyperpolarization. However, sometimes a number of positive ions will gather at the axon hillock, thereby making the inside of the neuron less negative than it typically is. Now, this is called depolarization, and it's the key to neuronal communication. So as noted before, the resting potential of a neuron is typically negative 70 millivolts. When a bunch of positive ions gather at the axon hillock, this value starts to climb up to negative 70 millivolts, then negative 65 millivolts, then negative 60 millivolts, and when enough positive ions gather at the axon hillock to push the polarization to negative 55 millivolts, then the neuron will fire. And when a neuron fires, it's called an action potential. So action potentials are an all or none occurrence. This means that once an action potential is triggered, when a neuron fires, there's no stopping it. Once it starts, it has to finish, and it never gets weaker. There's no such thing as a half action potential or a weak action potential. There's only a single all or none action potential. So we have an action potential. It's triggered when the neuron depolarizes at the axon hillock. And the action potential itself is actually the movement of this depolarization all the way down the axon. So, as you can guess, when it comes to communication in the brain, speed is going to be of the essence. And this is the reason for the myelin sheath. As noted earlier, the myelin sheath serves to insulate the axon from the external environment. This means that underneath the myelin sheath, no depolarization is required. In essence, the action potential can skip past any area of the axon that's covered in myelin. This means that an action potential doesn't have to travel smoothly all the way down the axon. Rather, it jumps really quickly across those nodes of Ranvier we saw earlier. Those little exposed areas between the myelin sheath. So this type of jumping signal is known as saltatory conduction, and it serves to drastically accelerate the speed with which an action potential can travel along an axon, because essentially there's a lot less ground for the signal to cover. Now eventually the action potential is going to reach the terminal button, and to see what happens next, we need to zoom in now to the synapse. When the action potential hits the terminal button, it opens up channels that allow calcium to flood into the presynaptic terminal. Now this calcium in turn pushes those chemical-filled vesicles against the edge of the presynaptic terminal, where they then release their neurotransmitters into the synaptic cleft. So inside the neuron, we had this informational signal that was electrical. But here at the synapse, that informational signal changes and it becomes chemical in the form of these neurotransmitters. When in the synaptic cleft, neurotransmitters don't really do much. They don't swim or propel themselves forward. Rather, they simply float around. So we call this now passive diffusion. And every once in a while, when passively diffusing, a neurotransmitter will bump into one of the receptors on the postsynaptic terminal. In order for the two to interact, however, they must fit together. So most neurotransmitters will not fit most receptors. So simply bounce into one another and nothing will happen. But occasionally, you'll get a match. A neurotransmitter will fit into a receptor, and when this happens, the receptor will open a channel, allowing the ions to flood into the postsynaptic terminal. And remember, ions are simply molecules with a charge. So for this reason, we call a movement of ions into the postsynaptic terminal a synaptic potential, and the informational signal now once again becomes electrical. So let's zoom back out and take a look now at neuron number 2. So remember when we started, we noted that ions will gather and fluctuate at the axon hillock, and it's this concentration of ions at the axon hillock that determines whether or not a neuron will fire. Well, those ions are the ones being led in by the receptors at the postsynaptic terminal. So ions are constantly flooding into the dendrites of neuron number 2, moving to and gathering at the axon hillock, and it's the sum total of all these ions that determines if the next neuron is going to be more negative and hyperpolarize and not fire, or become more positive, depolarize and fire. So it's worth noting, unlike the all or none action potential, synaptic potentials are graded and decremental. So graded means that the number of ions that flow into a dendrite from a specific synapse will always change depending on the amount of neurotransmitters and receptors within that synapse. So sometimes you're going to get a ton of ions flowing through one synapse, whilst other times you're only going to get a little trickle. Now, decremental means that the further away from an axon hillock a synapse is, the further these ions have to travel to reach the axon hillock, and the more that signal is going to deteriorate before it actually reaches the axon hillock. So this means that the further away from the axon hillock a synapse is, the less powerful that synaptic potential will be, and the less likely that synapse will determine whether the neuron will generate an action potential or not. So as we can see, with the incredible number of synapses sending in incredible amounts of ionic information, this can all grow incredibly complex. But if the resting potential of axon hillock of neuron number 2 hits negative 55 millivolts, then the cycle is going to start all over again. An action potential is going to be generated. It's going to travel down that axon, release neurotransmitters at the synapse, altering the synaptic potentials of the next neuron, and on we go. And this is how the brain communicates.